I now want to turn to the first speaker in this symposium today, and that is Jim Allison. And I have a special way to introduce Jim, if it plays. So let's see. Here is...
Part of Jim's success is the fact that he's an iconoclast. Great scientists are creative people.
Jim's lab has a feel of being like a pirate ship.
Jim theorized that tumors have evolved to trick the immune system.
At the time, it was a crazy idea.
This constituted a real breakthrough.
I knew that this is a way to cure cancer, and I'll be damned if I'm going to let that just lay around.
For the next decade, he had one goal, to get that made into a drug for cancer patients.
It was just no, place after place after place.
Many of my colleagues said, this will never work.
My mother sat on the bed with her and held her hand while she died, and found out later that it was lymphoma. When I was first told I had prostate cancer, I was terrified because it was a week after my brother had died.
BMS was not sure they were going to take it at all.
I must admit I was apprehensive.
His experiment in this was to just keep fighting, to say, there's something here.
He's willing to do it in the face of his colleagues not necessarily believing him.
He wanted to stay on the scientists, he wanted to stay on the FDA, he wanted to stay on Bristol-Myers Squibb.
I need to get it into people. I need to get it into patients.
I got a phone call from the radiologist. He goes, what did she get? All the cancer went away.
Until then, it was just a concept. She was the first patient I'd ever met.
How in the world are you supposed to adequately thank somebody that without them, you wouldn't be here?
That makes it all worthwhile.
It takes a really powerful idea combined with someone willing to push it forward to make it happen.
The number one thing I learned was how important it is just to persevere.
We knew we were changing the world.
May I ask you to step forward to receive the Nobel Prize?
This is a documentary
He's an iconoclast.
produced by Bill Haley. It's a pleasure to introduce the first scientist ever given a Nobel Prize for the cure of cancer. That's right. And the first scientist that I know of that played harmonica with Willie Nelson, Jim Allison.
Anyway, I'm not sure why I'm invited to this meeting on machine learning. I'm not going to really talk about that. I'll be talking about a lot of computational stuff that we've done in order to figure out the detailed mechanisms of action of anti-CTLA-4 and anti-PD-1, the two major checkpoint blockers that are in extensive use in treating many types of cancer now. Then at the end, I'll show you some conclusions that came out of that work that are really quite amazing and I think you'll find quite interesting. Anyway, there's my disclosures. The only one that's relevant is Jounce Pharmaceuticals, who I'm a founder of and who's, I'll mention when I get some stuff that we're doing with him. In any event, so just to briefly review where we are, this is one of my favorite slides. This is also a woman named Sharon, not the other Sharon Bellman, but this was a woman who was in the phase 1 trial in 2001. Let's see here. In 2001, and you can see this is what she looked like when she started on the trial. She had failed other therapies, including a dendritic cell vaccine, high-dose IL-2, a bunch of chemo, and had this large tumor mass and a pleural effusion. She got one injection of 3 mg per kg of ipilimumab, and about 4 months or so later, her tumors were completely gone. I visited Tony Rebus, who was her oncologist, 10 years after that in 2011, and she'd just come back for her first decade checkup and still completely tumor-free. Now it's 8 years after that, and she's still doing fine with just that one treatment, and that's where she is. So this just illustrates the durability of mobilizing the immune system to treat cancer. And so just to briefly review CTLA-4 and PD-1, CTLA-4 was the first cell-intrinsic regulatory circuit, negative regulatory circuit that was discovered on T cells by Jeff Bluestone's lab and mine. And the reason this is important in cancer is that tumor cells can't provide the necessary signals for co-stimulation. And so they're basically invisible to the immune system until they start dying in necrotic cell death. That causes inflammation. Antigen-presenting cells come in and pick up debris, and then redisplay it on their surface. Actually, initially, CD28 provides co-stimulation, but then in addition to turning on a GO pathway, which tells the cell to proliferate and differentiate, it starts an off pathway by inducing expression of the CTLA-4 gene. CTLA-4 accumulates, and we think gradually just begins to outcompete CD28 for co-stimulation. Just the cells then can't get co-stimulated again. So the main point from this is it works very early, at the immediate stage of priming of naive or memory T cells. There it is here. This slide's always slow for some reason. Anyway, this is a slide that was, it's about 4 years old now, but this was made when there was enough data for 10 years follow-up on 5,000 patients. And what you can see is that about 3 years, there's an inflection point, and the survival curve flattens out. And basically, what this tells us is that very few people are dying of melanoma after 3 years. After 3 years, they're basically good to go for 10 years and more, as I showed you with Sharon. PD-1 is very different than that. PD-1 is another checkpoint, works completely differently. And PD-1, one of its ligands is expressed on tumor cells, PD-L1. The tumor cell, the T cell, in its attempts to kill the tumor cell, starts secreting gamma interferon. That upregulates PD-1, PD-L1 on the tumor cell, which then turns the cell off. These cells are completely, fully differentiated effector cells. And so this is the opposite end of the developmental spectrum of where CTLA-4 works. Anyway, this is a trial that was done in 2015 where we combined anti-CTLA-4 with anti-PD-1, and the response rate is close to 60%. And the good news is the data now has matured out to 4.5, almost 5 years, and it's still there at almost 60%. So melanoma, just to remind you, when we started this work, the median life expectancy after diagnosis was about 7 months. And now we've got, looks like 60% of the people can last a decade or more. And so it's not just melanoma anymore. There's approvals in many different kinds of cancer. This is about 8 months out of date. And there's so many now, I just decided to stop updating the slide. But anyway, so there's a lot of effort in the field now, but the tendency is to say we've got 2 checkpoints, and what became clear really early on is that PD-1 has a slightly higher response rate, but the adverse events are much less frequent with PD-1 than with CTLA-4, so people say let's use PD-1, but they're very different. As I told you, CTLA-4 is hardwired. It plays a role in every immune response that you have. We know now that if you don't have its main function is to terminate immune responses. If you don't have it, your immune system will kill you. And we showed that in knockout mice, Ronnie Sharp and others. Whereas PD-1 is an induced resistance mechanism, which appears was initially had the role of protecting the fetus from attack by the maternal immune system on paternally encoded antigens. Anyway, it's thought that CTLA-4 targets the CD28 pathway, PD-1, the T cell receptor. So I told you CTLA-4 works during priming, PD-1 on fully differentiated cells. CTLA-4 has been shown to expand clonal diversity, that is, it can recruit new cells into a response. PD-1 does not do that. It just reawakens cells that are already there. Just going down, as I'll show you in a minute, CTLA-4 primarily affects CD4 cells, although there's also some CD8s engaged, whereas PD-1 only touches CD8 T cells. We recently showed in prostate cancer, Pam Sharma at MD Anderson showed that CTLA-4 can move T cells into, for example, cold tumors such as prostate cancer, which untreated looks like pancreatic cancer, but after a couple of doses of anti-CTLA-4, it looks like melanoma, and then can be treated with a combination of CTLA-4 and PD-1 antibodies, whereas PD-1 does not move T cells into tumors, from what we can tell. And One of the things that's appearing now is that disease recurrence after CTLA-4 is pretty rare, but it's pretty common actually with PD-1 monotherapy. There's about a 15% relapse rate in melanoma 2 years or so after the initial response. So they're very different. And reflecting these is the dosing. With CTLA-4, it's typically given 4 times at 3-week intervals and then stopped. And that's, despite that, people are still alive 10 years plus afterwards. PD-1 is given about every 2 weeks, depending on the disease and the company that's making it, about every 2 weeks for as long as 2 years and even longer. And I'll show you in a moment why that has to be done. So we wanted to really figure out, using an unbiased approach, which cells were affected by CTLA-4 and PD-1, and then by using CyTOF analysis, 39 parameters of transcription factors and cell surface markers for the major T-cell subsets, and then additional markers for proliferation, live, dead, other things, including barcodes so that we could analyze everything at the same time. And then we also kept track of the relationship and the number of different types of clusters of T cells with the size of the tumor, so we could actually relate changes to what's going on in the tumor. We studied 2 tumors, one MC38 tumor, which really is high mutational load, can be treated quite easily by both CTLA-4 or PD-1, very low myeloid cells. The other is B16 melanoma, spontaneous melanoma of mice, which used to be considered completely non-immunogenic. It's completely refractory to CTLA-4 or PD-1 unless you give a vaccine with it, a radiated tumor cell vaccine. I'm going to show you the data just for MC38, but I'll just tell you that the mechanisms are essentially the same, despite the fact that B16 is very low in mutational load. Like I said, it's essentially non-immunogenic, and MC38's got it quite at the opposite end of the spectrum. And so this is the scheme. We give the mice tumors. We can treat them with antibody, either anti-CTLA-4 or anti-PD-1 for this part of the talk. And then at day 13, which is when the tumor is just beginning to start shrinking, we harvest them, weigh them so we know the size, and then do the CyTOF analysis. So this is a phenogram. This, by the way, I should point out that this work, the computational stuff, was done with Donna Pear in her laboratory. And then with Spencer Way in my laboratory did all the work that I'm going to show you from here on. But anyway, these are the different clusters that you get of T cells when you do this analysis. And just to arrange them in a way that makes a little more sense, separate them into different cluster types of T cells. And so what we found in this example was in the situations there are 2 types of FOXP3 positive CD4s, or regulatory T cells. One of them has KLRG1, the other one does not. Both of these are associated with the tumors getting bigger, which you'd expect because they're suppressor cells. And this population is depleted by anti-CTLA-4. There's a decrease with PD-1, but we think that's dilution with other cells. But in any event, we have noticed this several years ago, as did Alan Corman, but this appears to be an artifact of the antibody that we use in mice. So far, we've looked at both antibodies to CTLA-4, ipilimumab and tremelimumab, which are in clinical use now, and we find no evidence in patients that there's any depletion of Tregs, so you can just ignore that for now. In the CD4 population, what you can see is PD-1 blockade doesn't do anything. CTLA-4 expands what we call a TH1-like cell. It's a TH1 because it has T-bet, it's this major transcription factor, and because these cells make gamma interferon and TNF-alpha, but they're unusual if they express high levels of this molecule called ICOS, or inducible co-stimulators. And this is very unusual, and I'll come back to this in a little while. And then among the CD8s, CTLA-4 also expands these cells, which are fully differentiated effector cells that are negatively associated with tumor cells. There's some other stuff, this cell population doesn't do anything, and then these that are also increased by CTLA-4, but they seem to get in the way. These seems to be tumor non-specific cells that are just cruising through the tumor. But anyway, just suffice to say that CTLA-4 hits these guys, and these are the important ones. PD-1, on the other hand, hits this population, these fully differentiated cells as well, but also expands this population of PD-1 high, TIM3 high, LAG3 high. These cells have 3 different immune checkpoints on them, checkpoint receptors on them, and so these cells are considered fully exhausted. But they are associated, these cells increase, and that's associated with decrease in tumor size, so they're important. But the thing is, they're still PD-1 high, LAG3 high, and TIM3 high, which tells us that if the antibody goes away, these guys are going to stop dividing. So, in any event, 2 different types of cells. So that may explain why they work together so well, but I'll come back to that in a minute. But suffice to say that there's different subsets that are engaged by PD-1 and CTLA-4. Well, what happens when you put them together is A plus B equal AB. So we just repeated this sort of analysis again using monotherapies or the combination therapy here and did exactly the same thing. And this is very interesting because what we find is these PD-1 high, LAG3 high, TIM3 high cells that are increased by PD-1 and they're involved in secreting, in tumor reduction, actually go down when you add CTLA-4 to the mix. They go way down. Now, this could be a reversion. Well, let me finish this first. But if you look at those terminally differentiated cells, just a plain old effector CD8 cell, these are increased by the addition of CTLA-4 blockade to the cocktail, much more than over what you get with PD-1. And so the loss of these and the increase of these suggests one of 2 things. One is that we're either converting these exhausted cells to not be exhausted anymore so they can proliferate, but I think that's unlikely because a number of labs have shown that these cells have epigenetic changes that locks them in that phenotype, which is why you have to give the antibody, again and again and again until the last tumor cell's gone. CTLA-4 increases them, and we think what's going on here is we take the inhibitory pathways off both when these cells begin differentiating, where they need co-stimulation, and prevent them from being stopped by PD-1, with the PD-1 antibody. And so these cells may not be, we just may avoid the generation of exhausted cells by providing co-stimulation. So a very different picture when you put the 2 together, which may mean, well, and this just shows that those exhausted cells, which with the monotherapies were clearly associated with smaller tumor size, also become irrelevant to the whole tumor rejection process when you add CTLA-4 as well. So this may mean that we could start thinking about maybe not having to give PD-1 as maintenance when you're doing combination therapy. We'll see, but it just shows a big difference. Anyway, on the CD4 compartment, what we see is a slightly bigger increase in those ACOS positive Th1-like T cells. So what are these cells? Well, ICOS is a close relative of CTLA-4 and CD28. It's very closely linked to the genome, highly homologous. It has its own ligand, ICOS ligand. If you ask an immunologist what an ICOS-positive CD4 is, they'll tell you it's either a follicular T helper cell that are in the germinal centers in the secondary lymphoid structures involved in TB collaboration and class switching of antibodies, or they're associated with a subset of FOXP3 positive regulatory T cells that make a lot of IL-10 and TGF-beta. But actually, they weren't strangers to cancer. Pam Sharma first found them in 2008 in a pre-surgical trial that she carried out in bladder, where she found out that there was a big increase of these cells both in the tumor and blood after IPI. These contain all of the tumor-specific gamma interferon and TNF-alpha producing cells. If you look at the ICOS negative cells, there's no action in there at all. with Jed Walchuk when I was at Sloan Kettering, we showed that the increase was associated with with longer survival. If patients didn't get that increase, they lived for a median of 8 months, whereas if they did get them, they lived with a median of 24 months or bigger. So we can't use that necessarily to predict who's going to respond, but we can use it to predict who's not going to respond. So if you don't get those cells, you just don't respond to patients. Anyway, it turns out to be a pharmacodynamic marker of IPI activity. In the mouse, Pam's lab and mine both showed that if you do CTLA-4 blockade in ICOS or ICOS ligand knockout mice, you lose about 2/3 of the efficacy. So it definitely plays a causal role in there. And this is attributed to a PI3K binding motif in the tail of ICOS that through the AKT pathway increases T-bet expression and makes these guys really super Th1s. And several years ago, we showed that you could target ICOS using, in this case, ICOS ligand expressing irradiated tumor cell vaccine. Whereas you can see here, if you treat just a mouse that's received a vanilla vaccine without ICOS ligand, you get about a 20% response rate. But if you have ICOS ligand in that vaccine, it goes up to bigger than 80%. And so that's associated with more tumor rejection. Anyway, so one of the things that we were curious about, because we did some early experiments, I noticed that there are a lot of ICOS positive cells in CTLA-4 knockout. So now we went to CTLA-4 knockouts and littermate controls, did a similar sort of analysis, and this is what it looks like. Red is knockout, and then blue and green are the wild type in the heads. So you can see these red areas that really don't appear except in the knockouts, including this Australia thing down here, where everything is, it's not connected at all with any of the rest of it. And if you look down there, what's in that cell, in that island, you can see these are not detectable at all in wild type or heterozygous animal. But there's our PD-1, our ICOS positive T-bet Th1-like cells, which again, we cannot find in human PBMC with our CyTOF panel, except in patients that have been treated with anti-CTLA-4. We cannot find those in lymph nodes in mice, except in mice which have been treated with anti-CTLA-4 or have had genetically the gene eliminated. And then we've got all these other guys here that are all mixed up. Like this guy has 3 major transcription factors, which should, associate with, this one, for example, with making type 2 cytokines, that with IL-17. So these cells are all messed up. And so we think what's going on here is that, T cells differentiate functionally after they've been activated, depending on what's going on in the environment. That's how you get all the Th1s, Th2s, and stuff. We think that when you remove CTLA-4, you open more differentiative space to the cells, which is that red, what I call the Australia-like thing. So, this just shows the old view of T cell differentiation, where if you get BCL6, your follicular T helper cell, GATA3, got Th2 that makes type 2 cytokines, T-bet, Th1 that makes gamma interferon, TNF-alpha, ROR, gamma T, et cetera, And this was thought to be, a linear process, again, determined by the environment that's going on. But there's been some data from John O'Shea and the late Bill Paul suggesting that's a more nuanced change, and there's some fluidity between the different types. Cells could change back and forth. And we think that one thing that may be going on here is that CTLA-4, by the way it works in countering co-stimulation, may effectively dampen TCR signaling strength. And when you take it away, you get supernatural, and I don't mean spooky, I just mean above natural levels of signaling. And we've known since Kim Bottomley's work in the late 70s that a stronger TCR signaling strength was associated with Th1s. And so we think then that the absence of CTLA-4 expands the phenotypic boundaries of the cells and giving you more types. And so one of the things that Donna Pear taught us is that there's clusters, which I've showed you. There's also archetypes, and these can be worked out by characterizing the neighborhood of the cluster. So you move slowly away, cells look at the most next closely related, then just further and further and further. And you could tell, for example, if you were going from here to here, did you just go that way, or was there, a more complicated distance, the maturation? And so if you look at the archetypes, this is the map you get. Anyway, this is Australia down here, and that's 7, cluster 7 here, archetype 7, again, are those ICOS positive Tibet TH1-like cells. And then these are the other ones that I mentioned earlier that are down here. So they're about as far as you can get from wild type or HETs, again. And so this tells us that, there is a differential path that's followed. The other thing that you can do is, of all the parameters, you can just track it as you march from, in this case, a naive T cell to the fully differentiated cells, and express that as pseudo-time. And this sort of shows the pattern here with archetype 7, where you lose stuff and then end up with other things. And what we know is, just ignore this for a moment, but archetype 7 starts out with GATA3, Tibet, and BCL6, all 3. And you can see here with developmental time, when they differentiate away from naive, they end up with just Tibet, they lose GATA3, and also lose BCL6, and that's the way the T cells go. And so with loss of PD-1, you don't really see that. You don't see anything in a PD-1 knockout that's not there in the wild type in HETs, even though the numbers change somewhat. So that might be expected, since PD-1 only affects turbidly differentiated cells. Anyway, what I've told you, I think, suggests that there's support for this nuanced model of T-cell differentiation, but also teaches us, I think, that when you're giving anti-CTLA-4, you're not necessarily expanding some cells that are already there. You're actually, because of these effects on differentiation space, it's actually creating the cells that are going out and killing the tumor cells. They don't exist until you give the drug, and they end up being, much more powerful killers than the standard CD4 TH1 cells that you have normally. And so with that, I'll stop. Again, all this work was done Spencer Way, and also collaboration with Pam Sharma, and then with Donna Perez's lab, starting actually when she was at Columbia. So I'll stop there. Sorry, I went over.